Good morning, everybody. Welcome back to Daily Lectures by New Age Cosmetic White, EdTech Healthcare Consultancy. We're on the subject of molecular biotechnology and have entered the fourth section, which deals with molecular biotechnology in society. And the topics in particular are regulating the use of biotechnology and societal issues in biotechnology. Right. So going ahead with the subject matter proper, let's just give an intro to it. Inevitably, new techniques and technologies, especially when they are widespread and pervasive as molecular biotechnology, affect society in diverse ways. There can be economic, social, and ethical consequences that result from both the implementation of a new way of doing something and the displacement of traditional processes. Moreover, because molecular biotechnology deals with the genetic engineering of life forms, it impinges on a number of socially sensitive issues. As a result, many questions have been raised about its propriety, safety, and acceptability to society. In part 4, we examined several of the controversial social, regulatory, political, and ethical aspects of molecular biotechnology. Regulating the use of biotechnology. The topics in this section are the subtopics would be regulating recombinant DNA technology, deliberate release of genetically modified microorganisms, regulating food and food ingredients produced by genetically engineered microorganisms, genetically modified crops, genetically engineered livestock, patenting biotechnology, patenting in different countries, patenting DNA sequences, patenting multicellular organisms, patenting and fundamental research, summary, and review questions. Now, major technological advances such as molecular biotechnology are seldom implemented without controversy. Because molecular biotechnology can potentially affect many aspects of modern society, including food production and medical treatment, there are significant ethical, legal, economic, and social issues that need to be considered. For example, since its inception in 1973, serious doubts have been voiced by some individuals about the safety of recombinant technology. And these concerns prompted scientists to declare self-imposed moratorium on certain types of recombinant DNA experiments until the adoption of official regulatory guidelines designed to ensure that recombinant microorganisms were unable to proliferate outside the laboratory and that laboratory workers were protected from any potential hazard. The formulation of these regulations took place in 1974 and 1975 in open meetings under the scrutiny of the press. Thus, the public became aware of the possibilities, both negative and positive, of genetically manipulating organisms. Nevertheless, in the late 1970s, apprehension persisted about the safety of this new technology. In particular, there was concern about the release, either accidental or deliberate, of genetically modified organisms into the environment and fear that they would become uncontrolled biological marauders of vulnerable ecosystems. Additional specific guidelines seem to be necessary to ensure that such rare possibilities would be even less likely to occur. In 1998, a vociferous and aggressive campaign was launched against planting genetically modified crops and marketing products derived from them. The reasons for this response are complex and encompass multiple concerns that range from human health and safety to environmental protection, corporate control of the food industry, world trade monopolies, trustworthiness of public institutions, integrity of regulatory agencies, and loss of individual choice. Moreover, there has been much discussion about the ethics of genetic manipulation of animals. The objective of these discussions has been to distinguish between inappropriate and acceptable procedures. There are no easy answers to the ethical, legal, and social questions raised by the various applications of molecular biotechnology. However, because the stakes are so high, many of these issues have been examined extensively. Regular recombinant DNA technology. In 1974, when it was realized that recombinant DNA technology, excuse me, could be used to engineer organisms with novel genes, concerns about safety, ethics, and unforeseen consequences raised by scientists, the public, and government officials phrases such as playing God, manipulation of life, the most threatening scientific research ever undertaken, and man-made evolution, were often used by the popular press when describing recombinant DNA technology. The major apprehension was that either inadvertently or possibly deliberately for the purposes of warfare, unique microorganisms that had never previously existed would be developed and would cause epidemics or environmental catastrophes in response to a certain amount of public anxiety about these doomsday bugs. A group of prominent molecular biologists called for a moratorium on several kinds of recombinant DNA research, especially experiments that involve pathogenic microorganisms. Subsequently, in 1976, the U.S. National Institutes of Health, NIH, the primary U.S. research grant agency in the medical and health sciences, issued guidelines for research involving recombinant DNA molecules, the NIH guidelines. These rules and regulations rigorously define physical laboratory containment levels for the conduct of recombinant DNA experiments. They also require that biological containment be a component of any recombinant DNA experiment, that is, the preferred host for foreign DNA would be those microorganisms considered least likely to proliferate outside the laboratory or to transfer the DNA to other microorganisms. For research with known pathogenic organisms, elaborate negative pressure controlled self-contained rooms were recommended. Research that was perceived to be less dangerous could be conducted in enclosed contained units equipped with high-quality filter systems. Although the NIH guidelines do not have legal status, most researchers, including those who did not receive NIH funding and private companies that were starting recombinant DNA technology research programs, voluntarily complied. In addition, other countries using the NIH guidelines as a model adopted their own sets of restrictions for the conduct of recombinant DNA research. The initial NIH guidelines were very stringent. Many scientists thought that they were excessive. For example, the cost of the containment facilities required by the guidelines effectively prevented smaller companies and researchers with modest grant support from initiating programs using recombinant DNA technology in anticipation of the need to modify the original guidelines. The NIH Recombinant DNA Molecule Program Advisory Committee was created. This committee was charged with overseeing the developments in recombinant DNA research and, if necessary, refining the regulations. The RSC had to hold open meetings to discuss its decisions. It had to publish and distribute the minutes of its meetings, and it had to allow any non-member the opportunity to address the committee on any issue pertaining to recombinant DNA research. The membership of the RSC was broad and included both ethicists and members of the general public, although it consisted mostly of scientists. As part of the original NIH guidelines, one specific class of experiments that was not to be initiated at the present time under any circumstance was the deliberate release into the environment of any organism containing a recombinant DNA molecule. However, it was inevitable that genetically modified organisms that could function in natural settings would be developed. By the 1980s, The original NIH guidelines were relaxed considerably by the RSC as a result of experience and specific experimental data from studies that committee and NIH had sponsored. For example, it was established that the host organism, E. coli K12, which was most commonly used in recombinant DNA experiments, was unable to proliferate to any significant extent outside the laboratory. In addition, microbiologists convinced molecular biologists and others that existing safety measures for work with pathogenic organisms were of high standard and that more stringent rules were not required. Finally, it was considered that it was extremely unlikely that a pathogenic organism would be created if the cloned gene had nothing to do with the pathogenesis in the original organism. Most observers were satisfied with the safety of laboratory workers was ensured if good laboratory techniques were used. However, specific regulations were added, the guidelines to safeguard against accidental spills during large-scale fermentations with genetically modified organisms. I'm just going back to read the milestone note is about potential biohazards. Potential biohazards of recombinant DNA molecules. Courtesy and gratitude to P. Berg, D. Baltimore, H. W. Boyer, S. N. Cohen, R. W. Davis, D. S. Hognes, R. Roblin, J. D. Watson, S. Wiesman, and N. D. Zinder in Science 185, 303, 1974. Thank you to them for this wonderful note. After Cohen et al. In Proctor National Academy of Science USA in 1973 described the basic strategy for inserting a DNA molecule from any source into a plasmid. The safety and ethical implications of recombinant DNA technology were discussed at length in the scientific community. These informal discussions led to the formation of a temporary committee, ad hoc committee on recombinant nucleic acids that was composed of leading molecular biologists who were charged with examining the concerns in more detail. In 1974, this group published a brief note which was dubbed the Berg Letter, simultaneously in 3 major scientific journals, Science, Nature, and the Proceedings of the National Academy of Science of the United States of America. In this communication, Berg et al. Recommended that researchers defer creating microorganisms with novel genes for drug resistance and toxin reduction and cloning cancer-causing genes from viruses into bacterial host cells. Moreover, they suggested that a government body, which would represent the interests of the public, should prepare guidelines and regulations for recombinant DNA technology and that an international meeting of scientists should be convened to discuss appropriate ways to deal with potential biohazards of recombinant DNA molecules. This letter had a significant impact. It led to a scientific meeting at the Asylum Conference Care Center in California that attempted to define various recombinant DNA experiments in terms of their relative risks and suggested which laboratory safeguards should be implemented for experiments that were considered to have a minimal, low, moderate, or high risk. In turn, the Asylum recommendations, Berg et al. Contributed to the development of the 1976 NIH guidelines that established stringent regulations for all types of recombinant DNA experiments. The Berg letter was responsible for the formulation of regulatory framework for recombinant DNA technology. It focused public attention on the potential risks and benefits of recombinant DNA technology and ignited a heated public debate about the technology. Historically, it represented an interesting example of scientists trying to anticipate the possible risks of a new technology before there was any substantial evidence that there were any hazards. As a result of the easing of containment requirements for more routine experiments, the use of recombinant DNA technology became more prevalent and flourished. The RAC and the NIH guidelines had effectively quelled to some extent the original concern about the potential dangers of recombinant DNA research. However, two significant problems remained. First, what kind of regulation was needed for foods that contain either genetically modified organisms or products that were derived from genetically modified organisms? Second, how were deliberate releases of genetically modified organisms into the environment to be managed? A third potential problem area was avoided when pharmaceuticals that were made by recombinant DNA technology were deemed by regulatory authorities to be similar to pharmaceuticals produced by traditional means. In most countries, there was a strong consensus that existing regulations for the approval of pharmaceuticals for commercial use were sufficient to ensure both worker safety and public safety, and that the process, that is, recombinant DNA technology by which a product was made, was irrelevant. The axiom that the product alone should be evaluated for its safety and efficacy led to the approval of a range of recombinant DNA products, including human drugs, vaccines, and diagnostic devices. Deliberate release of genetically modified organisms. Despite its initial prohibition by 1982, it became clear that the RAC would have to cope with requests for open field testing of genetically modified organisms. That is, for the deliberate release into the environment. Uncharacteristically, neither guidelines nor protocols that advised applicants what information should be included in their submission had been prepared. This initial reluctance to establish definite regulations was due to a widely held belief among molecular biologists that genetically modified organisms were not significantly different from their non-engineered progenitors. And if a difference was present, it was thought that it would be readily detected by conventional biological testing. 3 applications for field trials of genetically modified organisms were received by the RAC in 1982. 2 dealt with genetically modified plants, corn, maize, and tobacco. The third proposal was concerned with testing a genetically modified strain of the microorganism Pseudomonas syringae to determine if it could limit the extent of frost damage to plants. This particular submission became part of the landmark case for the development of regulatory procedures for the release of genetically modified organisms into the environment. The genetic engineering portion of the Pierce-Syringay proposal involved removing a gene that coded for an ICE nucleation protein from the organism, and then testing whether the modified ICE- strain when sprayed onto the leaves of plants could prevent frost damage. Under natural conditions, wild-type ICE plus Pierce-Syringay, which is usually found on the surface of plant leaves, secretes a protein that at low temperature causes the formation of ice crystals, which in turn causes frost damage to the plant. The rationale for the deletion of the gene encoding the ICE nucleation protein was that if a strain that lacked this protein was sprayed onto leaves before they became colonized with the plant wild-type strain, it might lower the temperature at which ice formation would occur, thereby preventing the leaves from being damaged by bacterially induced ice crystals. There is a significant economic incentive for such a novel treatment because in the United States, crop losses due to frost damage cost farmers billions of dollars each year. In response to each of the requests for field testing of genetically modified organisms, the RIC followed more or less the procedures it had established for handling the regulation of recombinant DNA experimentation in the laboratory. The submissions were announced in the U.S. Federal Register. Information was sent to 3,000 interested persons. A panel of experts reviewed the proposals. A public meeting was called for discussion of each proposal. At the same time as the RIC was reviewing the proposals, the U.S. Department of Agriculture, USDA, also reviewed them. After careful consideration, both the USDA and the RIC approved the ICE nucleation gene deletion proposal in 1993. The director of NIH gave final endorsement of the RIC decision. On the same day that the permission was granted to proceed with the field trial, a lawsuit to block the test was filed by an organization called the Foundation on Economic Trends which is headed by Jeremy Rifkin, who strongly opposes all forms of genetic engineering. The lawsuit was upheld, with the judge noting that the RC had not carried out a proper hearing in accordance with the US statutes and, more importantly, that it had failed to request an environmental impact statement. This release of a legal decision dramatically demonstrated that despite the scientific opinion of the RC and its experts, existing regulatory system for field testing of genetically modified organisms was inadequate. Prepared an opinion outside the confines of the RC was that the release of a genetically modified organism into the environment could have far-reaching effects because living microorganisms proliferate, persist, disperse, and sometimes transfer the DNA to other organisms. Some critics of the release of genetically modified organisms into the environment believe that after its introduction into the environment, an engineered organism could displace an existing important species from its ecological niche and as a result cause severe environmental damage. In addition, some opponents of release believe that genes could be transferred from an introduced genetically modified organism to indigenous strains, thereby creating albeit inadvertently an ecologically dangerous organism. Although these points of view present a worst-case adverse effect scenarios that might be exceedingly unlikely, it was essential that the regulatory protocol for field testing include a thorough assessment of the potential risk that an introduced organism might pose for the environment. The responsibility for assessing the initial submissions for the deliberate release of genetically modified organisms in the United States resides with the U.S. Environment Protection Agency, EPA, and USDA. The NIH drew up an initial set of criteria for field tests with genetically modified organisms, but it relinquished its authority in this area to these other agencies. The EPA decided to use two applications, both dealing with the ice nucleation defective bacteria as prototype case for developing an assessment process for the field testing of genetically modified organisms. Each proposal went through a series of reviews, which include appraisals of the environmental fate, ecological effects, and human health consequences of the test, as well as product analysis by the following groups: the Office of Pesticide Program Review of the EPA, the Toxic Substances Research and Development Policy Planning and Evaluation Committees of the EPA, the General Counsel of the EPA, USDA, FDA, NIH, Science Advisory Panel that consists of microbiologists, a plant pathologist, and a community ecologist, open public meetings, various state agencies, which in this instance included the California Department of Agriculture. It was not envisioned that this elaborate, time-consuming, and often redundant process would become the routine mechanism for approving field testing of genetically modified organisms. Rather, it was assumed that with experience, the system would be trimmed without loss of effective assessment of potential hazards of each trial. After what was thought to be a very thorough set of analysis, permission was granted for both of the field trials with the ice nucleation negative bacteria. However, in both instances, although the circumstances were different, local residents who were worried about the release of genetically modified organisms in the neighborhoods obtained court orders that temporarily blocked each of the field trials. As a consequence of this delay, both the EPA and the USDA implemented better methodologies for determining the risks of introducing genetically modified organisms into the environment. In a short time, the staffs of these agencies became more proficient at handling and analyzing the data submitted by applicants. The scientific community, including ecologists, helped the process by initiating research programs that were designed to examine the consequences of the release of organisms into model environments, and scientific organizations formulated frameworks for deciding whether a particular genetically modified organism would have an adverse effect on the environment. Eventually, in 1987, the field trials with ice nucleation negative bacteria were conducted at sites in California. The results indicated that these genetically modified organisms were not dispersed to off-site locations, nor did they persist at the site of application. At one site, the freezing temperature of the test plants were lowered by 1 degree centigrade. However, for a number of reasons, genetically engineered ice-minus bacteria have not been developed commercially to protect crop plants from frost damage. Since the first trial device managed bacteria, open field tests of genetically modified microorganisms have become commonplace. Overall, these studies have found that introduced microorganisms tend to remain confined to the test area, do not persist for more than a few months, do not transfer genes to indigenous organisms, microorganisms, and have similar basic biological functions in natural laboratory settings, generally because there can be many differently possible adverse effects for each genetically modified organism. A case-by-case approach has been adopted by granting permission to conduct a field test. These kinds of tests have been carried out in the United States, UK, Australia, and other countries. However, biotechnology companies have been reluctant to develop genetically modified organisms that can be used in the environment because the cost of the field testing is high and because final approval may be denied despite successful test results. Nevertheless, there is a growing consensus that the environment release of genetically modified organisms after the appropriate laboratory and field tests will not be ecologically deleterious. Excuse me. Regulating food and food ingredients. In the United States, the FDA is responsible for regulating the introduction of foods, drugs, pharmaceuticals, and medical devices into the marketplace. The safety of both crop foods and food ingredients that include flavors and additives must be thoroughly assessed before they can be licensed for human consumption. The FDA has had a well-established, although not foolproof system for the approval of new foods and food products for some time. Critics of the FDA, however, have argued that it tends to favor the interests of industry and is too lenient in enforcing its own regulations. Both the FDA and the food industry, which is represented by the International Food Biotechnology Council, have argued somewhat convincingly that the new regulations are not required for foods and foodstuffs that are developed by recombinant DNA technology because any unilateral food or food ingredient, regardless of how it is produced, must be assessed for safety by toxicity, allergenicity, and impurity testing. The approach in the United States has been that food products produced through recombinant DNA technology are not considered to be inherently riskier than those derived through traditional forms of genetic improvement, such as selective breeding or cross-pollination. In other words, new food products arising from the genetic improvement, regardless of the method used, should be evaluated for risks, not the method by which the product was generated. Commercially available food products derived from genetically modified organisms are derived as safe as those derived from their non-engineered counterparts. Food ingredients produced by genetically engineered microorganisms is the next topic. We talk of chymosin. A new food product is usually subjected to a large battery of tests. However, in order to streamline the process and lower the cost of developing a food product, the similarity of the new products to the one that is designed to replace is taken into consideration. For example, the FDA approved the enzyme chymosin, an agent produced by recombinant DNA technology for use in cheese making, without demanding a full range of tests. Chymosin, one of the key components of RNA, is a milk-clotting proteolytic enzyme that hydrolyzes the casein protein in milk. This enzymatic cleavage creates curds, which in turn are processed into cheese. Traditionally, the milk-clotting agent for cheese making is derived from the fourth stomach of calves and consists of a mixture of substances that collectively is called RNA. To ensure a reliable, convenient, and possibly cheap industrial supply of chymosin, one of the chymosin genes was cloned and expressed, and the product was harvested from E. coli K12. This lecture will end in a couple of seconds from now and will follow up in the next lecture, right? And please take notes and screenshots, and we'll be back with you in the next lecture on the same topic. Thank you. Good day. The additional topics mentioned will be mentioned in the next lecture. Thank you. Bye.